What we're going to do here is highlight using Oncomine power tools to take advantage of the TCGA data and other available cancer genomic data to answer important cancer drug development questions. So the first case study that we're going to do here is a simple one. The question is, summarize my gene's profile across all available genomic data, including all of the data that's been collected from the Cancer Genome Atlas. And the particular questions of interest are, is my gene of interest mutated in cancer? If so, does the gene tend to be mutated at hotspots, which might indicate that the gene is oncogenic? And if so, at what frequency and in what particular cancer types? Similarly, is my gene of interest amplified? Are the amplifications focal and high level? Again, if so, in which cancer types and at what frequency? And then finally, is my gene significantly overexpressed? Is it overexpressed broadly across cancers or in particular subsets of tumors? So we're going to focus the case study on HER2 as we consider additional opportunities for anti-HER2 therapies. As we all know, HER2 amplifications and treatment with anti-HER2 agents really is a model for targeted therapy and biomarker-driven drug development. As I'm sure everyone's aware, HER2 agents such as trastuzumab or Herceptin and lapatinib are approved for HER2-positive breast cancers. As you likely know as well, more recently, Herceptin has been approved for gastric cancers, as gastric cancers also have high-level HER2 amplifications in subsets of the population. So what we're going to do in this case study is ask the question, does the global collection of genomic data, including the Cancer Genome Atlas, suggest that HER2 therapies are best directed at breast cancer and gastric cancer? And more importantly, does the data suggest other equally interesting indications that could be targeted with anti-HER2 therapy? So specifically, are there other cancer types or subtypes that have equally high-level focal amplifications of HER2? And asking the question from a somatic mutation perspective, do hotspot mutations occur in HER2 in particular cancer types and subtypes? And do we see significant overexpression of HER2, perhaps in cancers that don't undergo a high-level amplification? So that's the case study. And to investigate this, we're going to use the Oncomine power tool called the Integrated Gene Browser. This is a tool, as Matt said, that we just recently released. What I've done here is simply a search for HER2, and our first view on the HER2 genomic profile are these summary bar charts here, summarizing at the top the mutation frequency of HER2, then the copy number frequency, and we're filtering cases that have greater than an estimated 4 copies at the HER2 locus, and then significant overexpression frequency, and we're filtering on cases that have tenfold overexpression of HER2 relative to the baseline. So we're summarizing here all available mutation, copy number, and expression data for HER2 across tens of thousands of patients, including thousands of patients from the TCGA. So because HER2's approval, anti-HER2 therapies approval, are really focused on HER2 amplified cancers, let's first look at the copy number data for HER2. When we set it at greater than 4 copies, what we can see is that HER2 is amplified most frequently in breast cancer. As I'm mousing over this data, you can see that our estimated frequency is 13%. I think that's a little lower than what's been reported, but this is the value that we get when we look across 645 breast cancer cases and find 83 amplifications at our threshold. The data suggests that in gastric cancer, the frequency is nearly the same, 12% frequency, with 20 amplifications being observed across 171 cases. What I'll point out here is that HER2 amplification frequency is not 0 in all other cases. Instead, we see HER2 amplifications occurring at a 1% frequency in colorectal cancers, 8 out of 600 cases. Similarly, in lung cancer, a 1% frequency with 13 amplifications out of nearly 1,000 cases. as well as in a couple of other indications. So let's look at the actual data here. So now we're going to drill down on the actual cases that showed HER2 amplification. So I've moved over to the copy number tab. What I'm showing you now are data on nearly 10,000 cases. We're looking at HER2 copy number across 10,000 cases. So each circle is a case. We're organized by cancer type, and we have estimated copy number on the x-axis with our 4 copies threshold highlighted here. And what you can see is exactly what the frequency data told us. Frequent amplifications in breast cancer, frequent amplifications in gastric cancer, but rare amplifications in other cancers as well. So we see here in lung cancer, 1.2% with 13 out of 1,000 cases. When we click on lung cancer, we get details on the particular types of lung cancer that have been profiled. And what you can see is that the amplifications have most frequently been observed in non-small cell lung carcinomas, and specifically those designated as lung adeno and squamous. In a small number of small and large cell cases, no amplifications were observed. I'll also click here on the other cancer where we have 5 amplifications observed. A number of cancers represented here, most notably endometrial serous adenocarcinomas. While the N is low, only 14 cases, we're seeing 3 amplifications of HER2 in these endometrial serous adenocarcinomas. So perhaps with an expanded cohort, an interesting population for anti-HER2 therapy. But as we mentioned earlier, colorectal and lung cancer showing approximately 1% of cases. And given that these are large populations, these may represent new small indications for anti-HER2 therapy. Similarly, about 1% of ovarian cancer showing amplification as well. And for any of these cases, we can select the cases and get all of the details, the particular data sets, information about the samples that showed amplification here. Now the question is, though, are these amplifications in lung cancer likely drivers of these 1% of lung cancers? In breast cancer and gastric cancer, we know that these amplifications are relatively focal and center around the HER2 oncogene. What we can do now with our DNA copy number browser is ask this exact question. So what we're going to do is we're going to select lung cancer data sets for which we have copy number data. And I'm showing you here 4 data sets that have the greatest number of lung cancer specimens. So you can see Weir lung has 370 cases. The TCGA lung study, which was recently incorporated, adds 170 cases to this query. And what we're going to do is we're going to examine here the lung cancer genome in this top panel. So this is all genes in the genome ordered by chromosome position. Chromosome 1 genes, chromosome 2 genes, chromosome 3 genes, etc. And what we're doing here, somewhat like the GISTIC algorithm that you may be familiar with, what we're doing here is we're summing log2 copy number to point us to the regions of the genome that have frequent and focal amplification or deletion. So I've got HER2 highlighted here on chromosome 17. Let's zoom in to chromosome 17 to take a closer look of this region. So what it looks like is we have a region of the chromosome that shows relatively increased amplification relative to the surrounding area. And what we can do is we can select this region and ask the question, is HER2 the likely driver based on the focality of the aberration? So let me zoom in a bit here. So now what we have, the rows are cases, and you can see TCGA cases here at the top of the map, but cases from other studies as well. The boxes indicate gene-level amplifications. So the boundaries for a particular patient indicate the boundaries of the underlying amplification. So we can look across all the patients here. You can see the HER2 gene here in the middle. For the most part, HER2 is always amplified when the region is amplified. This trace along the bottom here summarizes the number of cases with amplification of the particular gene in blue and the degree of amplification in pink. And by examining these traces for the peak, you can find the genes that are most likely the driver. And indeed, we see a 3-gene peak here spanning from HER2 to GRAB7 that is likely the driver of these amplifications occurring in lung cancer, suggesting that much like gastric and breast cancer, HER2 amplification in 1% of lung cancer may indeed be a driver of the disease. We could do similar investigations in the ovarian cancer data, the endometrial data, etc. But I think what this data suggests is for anti-HER2 therapies that there may be really a sizable number of patients that could benefit from anti-HER2 therapy across the 1% of colorectal, lung, and ovarian. So I think something to consider for HER2 therapies being developed today. Okay, so let's move on now and consider mutation data. So we'll ask the question, does HER2 undergo mutations in cancer? And specifically, do those mutations occur at hotspots, which suggests that the mutations are likely oncogenic drivers? So what I've got here now is a summary of all of the mutation data that we have for HER2. The table on the top left here shows you the number of cases that we have tested for HER2. And again, this is a compilation of data from COSMIC, from the TCGA, and other next-gen sequencing studies. And we're sorting by frequency of mutation. What you can see here is for most cancers, we only have a small number of cases with mutations observed, although multiple recurrent mutations. What I'm focusing on here is in lung cancer. So we just went through the exercise to say that approximately 1% of lung cancers have high-level focal amplifications of HER2. What this data suggests is that approximately 1% of lung cancers also have a mutation of HER2, 64 out of 4,800 cases. Down in this bottom table here, we're again breaking this down by lung cancer type. What you can see is that 2% of the mutations are annotated in cases to be lung adenocarcinoma, whereas only 0.6% are annotated in squamous cell lung carcinoma. So perhaps an enrichment towards lung adenocarcinoma, similar to how KRAS mutations are enriched in adeno relative to squamous. Now, importantly, though, the question is, are these mutations occurring across the full spectrum of the protein, and thus we might worry that they are passenger mutations, or are these mutations occurring in particular hotspots? The pie chart here on the right indicates the amino acid changes that have been observed in HER2 across these 64 mutated patients. And what you can see from this pie chart is a few large pies indicating particular amino acid changes that occur disproportionately relative to other sites. What you can see here is that we have 19 cases representing 30% of HER2 mutations that are this particular amino acid 774 to 775 insertion, and 18% of the mutations are an insertion just one amino acid down. And as it turns out, these are similar to the EGFR insertion mutations that are known to occur. So we're summarizing data here below, and for data that COSMIC has curated, we can always drill in and look at the particular studies that have made the observations. In the case of TCGA data, you could drill into the data further in our TCGA tools. So with that, I think we'll conclude the HER2 case study to say that in addition to breast and gastric cancer, HER2 is rarely shows high-level amplification in colorectal, lung, endometrial, ovarian, and others. And is also mutated rarely across cancer types. In lung cancer specifically, it looks like those are driver oncogenic mutations based on the hotspots. Could be passenger mutations, but those mutations that we're most interested in are novel oncogenic mutations that are activating the protein and are potentially amenable to therapeutic intervention. So what I'm showing you here, much like what we saw for HER2, I'm showing you the mutation spectrum of two different genes, BRAF in melanoma and a gene called Titin in ovarian cancer. And I would just